Good morning, sir. Good morning, everyone. Today, our topic is deep learning in healthcare. Deep learning involves majorly image and text processing in healthcare. It has other application in different domain like business, satellite picture, environmental analysis. A lot of variety of domain is there, but we will focus mainly with our healthcare domain. So, grossly, the topics is machine learning and radiomics analysis. Already, we have idea about the machine learning techniques, supervised and unsupervised mainly. And there is a whole area of analyzing medical images. Images may be different types. That may be radiological image, histopathological slide image, molecular image, or different any other modality that can be think of. So that image is analyzed through AI techniques. To separate processes there. One is radiomics and machine learning, another one is deep learning. So we will first see radiomics and machine learning and how deep learning has introduced a whole new era to it. And then we will go for the text means the unstructured text analysis using deep learning techniques. And the basic building blocks of deep learning, of course, the neural network, how what is it is and how it is trained for any kind of analysis. So first, we'll start with radiomics and machine learning workflow, where first step is having the image with pre-processing of all kind of normalization, noise reduction, and annotation. Annotation can be of any kind like benign or malignant for screening, or follow-up data means responder or non-responder, or any type of disease identification, detection, the area demarcation. So annotation can be of anything. So first is image acquisition and pre-processing. Next is image segmentation. When I am talking about segmentation, that means annotating the exact area where the machine learning should look into. That may be a tumor, that may be a area where fibrosis is developing, or any kind of anomalies that is different from the healthy anatomical tissue structure. For that purpose, to direct the machine learning model to look into the specific area, the ROI, the region of interest is annotated. That is called segmentation. That may be manually done, that may be programmatically done, semi-automatic or fully automatic. These are all whole different computer vision areas. How images are segmented using automated or semi-automated method. Next is the feature extraction. We now have the exact area in the image, and now we have to collect different representation of the area. That representation can be shape in 3 dimension. Think about a lung nodule, or think about a HCC developed in the liver. So in 3 dimension, how the area has been proliferated in the anatomy, that total in 3 dimension, that gives the shape of that anomaly, that pathological structure. So okay, so I'm talking about shape. So shape can be of different uh, inference. When we are looking into cellular image, the cell structure outline of the cell may be information to classify the different cells. So who are from molecular biology or cellular biology, they can understand how cellular structure can differ in terms of shape. Then for tumor malignancy or the proliferation of tumor, how the boundary line is looking in the radiological image, that tells a lot of information about the malignancy or benignness. So shape is also a very important feature, how the clinician perceive the characteristics of the anatomical part or tissue or cell in the image when they look. And again, there are intensity level. In any modality, there is intensity. That may be Hounsfield unit, that may be in MRI gray levels. For different type of MRI images, the intensity tells about different level of quantitative information, like different diffusion coefficient or perfusion coefficient. So there may be lot of information in intensity. So how is the intensity profile inside that segmented area? That tells a lot. And then the textural features means how the spatial orientation of the gray levels in the image. This is a important computer vision area where human perceive how the human perceive, that is to some extent we can perceive a image, but how the pixel level gray intensities are oriented and how the cross-talking between the pixels, that human cannot perceive, and that may be formulated using the computer programming. Here that texture and of a area and wavelength based texture analysis, this is very important to identify a characteristics of a tissue in the ROI. So these are the whole domain of feature extraction that can be done using programming. And these features can be used for model development for different objective like screening, diagnosis, treatment, follow-up, decision risk stratification, etc. So along with our clinical features that already we are very well-known things, demographic, clinical features, and dietary habits, anthropometric measurements, all are our very common clinical parameters that we use for this type of clinical decision making. Along with this, when image comes into the picture, all these features are added to it to have more precise diagnostics. And here the machine learning comes into the picture with all these radiomics features that can enhance the decision making. So in this flow, the radiomics, what it is doing, it converts image into mineable data. When we are seeing an image visually, with our experience and our expertise, we can perceive how the pathology has changed, how much tissue has been corrupted by the disease. All these are our experience, our expertise. Machine don't know anything. They have only the gray level pixel values. Just using the mathematical formulas and the shape and the orientation, textural parameters, these are all mathematical formulas. And this, what we are perceiving in our brain, machine is perceiving with that calculations. So that is the radiomics domain that we already have talked about, the different type of radiomics features. So it is a mineable data, means it is a quantitative value. Now in terms of feature, that now can be used for the machine learning model for supervised learning. So it is a valuable information where machine don't know anything about the spatial orientation and anatomical structures. So they only understand the value. Now, the shape can be of different things. Intensity profile may differ, and the texture within the anatomy can differ. That we can understand by our experience. So now in this whole process, I'll share some research from my previous studies. So for example, if this is a T1 value, absolute T1 value in MRI images, it is a quantitative value in tissue where it differs. When we see the intensity, means it is a cross-sectional image of our thigh, means it is in the extremity in cross-section area of thigh, where red outline is in the tumor part. Here the tumor part is here. And these are all healthy tissue levels. So intensity profile means how the intensity is distributed, the values. In the x-axis, it is 500 to 1500. It is showing, and y-axis is the number of pixels. This is called histogram, the intensity distribution of the ROI. Simply to looking to the histogram means the profile of intensity distribution in the ROI. You can simply tell the red one is tumor and the blue one is healthy. The pattern, the shape of this histogram is itself very different. So only this much analysis will be enough to identify whether the area is tumorous or not. And with that, we can also identify chemotherapy response, who will be the responder and non-responder if we see baseline and follow-up intensity distribution. In baseline, tumor histogram was looking like this. It is a normal distribution. And in follow-up, it is skewed positively. In non-responder, the skewness is not that much. It was normal at baseline, both responder and non-responder was similar. But after chemotherapy, the profile has changed. So during the follow-up at early stage, when just one chemocycle is given, we can identify, at least predict to some extent, how much the probability is to response in the patient. And accordingly, that treatment can be changed, means personalized treatment. So it was the accuracy area under the curve was 0.8. So it's a quite good accuracy level, how we can identify just merely seeing the intensity profile in the tumor at baseline and follow-up. Another example is with correlation with the histogram analysis. This is the apparent diffusion coefficient parametric map. This comes from the diffusion weighted MRI, where this tells about the how much diffusion, water diffusion in cellular level is there. So this is the blue outline is showing tumor area, and just the intensity profile can tell about the how much responder and non-responder is having the necrosis area. It is a contrast enhanced MRI. The red outline is showing the contrast enhanced MRI, where dark area is the necrotic area. And in diffusion weighted MRI, it is quantifying the exact diffusion profile and how much diffusion profile is we can use as a marker for response with correlation with histopathological image. Again, in prostate cancer, the segmentation of prostate different area, peripheral zone and the central zone, and the textual features analysis can tell about the grading. Because this is a histopathological subjective process to grade the prostate cancer. We can use the texture features to have automatize the process, removing the subjectivity to some extent and having the grading of the prostate cancer. This is the example. This can be applied for fibrosis grading, HCC diagnostic and prognostics also. So similar type of structure, the process, workflow is the same. Application may vary. So from our domain, if it is cellular imaging or histopathological area or radiological imaging, the same workflow can be followed for clinical applications like diagnosis or follow-up or risk stratification or grading. So this was the radiomics analysis and machine learning method for clinical application using medical imaging. So what was the whole picture? The image acquisition, segmentation, feature extraction, intensity and different texture, and then feature selection, who are the best for the application, and then using the data analysis, the model and the disease prediction. So this whole lot thing is very time consuming, and the ROI segmentation is subjective. You are doing manually, or if you are doing by programming, it is maybe erroneous. So And the whole process, if we think about each and every process, it takes lot of time, human time, computer computation time. So this is a useful process. It gives lot of explainability and understanding how the model is picking up the feature and what features are going for the model to identify exact application, giving good result. This is very explainable and understandable, clinically useful, but long time consuming and long processing time is there. And there is another problem. When very similar type of things problem is there to classify, like classifying dog and cat, both have tail, four leg, 2 ears, similar type of features of face. Then if we have to differentiate in between this type of thing, where both class are quite similar, but minute change is there, that we understand in our brain, but still it is difficult for computer vision. That is also difficult using radiomics, that feature and shape, that is also difficult. So then this deep learning can come into the picture. It replaces all the feature detection and feature engineering part. The feature engineering is the term of having segmentation, then feature extraction, then feature selection. Everything is combinedly called feature engineering. So that feature engineering is replaced by the deep learning. So here the importance of deep learning for image analysis, you can understand the whole process. All these 5-6 steps is now accumulated in this neural network process. Everything is now accumulated here, and we have the output at the end. Of course, everything is inside a neural network process. So it is a black box. It is not very good clinically explainable and not very understandable what is going on in this black box neural network. So there is some pros and there is some cons. Everything comes with plus and minus. So in this deep learning, the long history is there. So small neural network to complex deep neural network to image analysis, deep learning in CNN, and then transformers that handles text, image, any kind of data. That is the basic foundation of our generative AI. So this is a whole long history. So now we will start with the basic building blocks that is called neural network. How it handles data. When there is very complex data in the area, how it is handling the non-linearity in the data. So the intelligence is the capacity to learn and solve the problem. In our biological neuron system, the dendrite in the neuron body and action is there. So dendrite is for capturing the information, processing the data is done in the neural body, and how the action is taken, that depends upon the neuron response. Means if it will fire or not, that takes the decision is taken in this neuron body. So it is a binary processor, either the neuron will fire or not fire, and the action will go through the action path. So analogy to this structure now come to the artificial neuron, where the dendrites means that input are coming through this X1, X2, X3. These are all input flow of input to the artificial neuron. Neuron body is this summation signal. Summation means this is a notation for mathematical summation. Means this input X1 will be multiplied with W1, will be added with X2 multiplied by W2, plus will be added by X3 into W3. Every input have some weight. Weight means the significance level for our ultimate output. The coefficient, you can remember the logistic regression coefficient. Here the weights are associated. Yes, this is the input features. We are giving input like dendrite. The input is coming to the artificial neuron body with their weights, means importance. It is some summation is done. The bias is the variability coefficient you can tell about. So this is also added. Now this is processed. The value is processed. Whatever the value is, the activation function is the similar as the action potential path, means the neural body, how it decides it will fire or not. The activation function is the similar kind of thing. It is positive response or a negative response? It defines. Mostly it is a sigmoid function that we already learned. The value is transferred to a 0 to 1 probability value, or it can be other type of activation function. Depending upon different activation function, ultimately some value goes to the output. Most of the time it is from 0 to 1 value, and it is 0.5 above is the positive class and 0.5 below is the negative class. This is the conventional idea. So this whole summation and activation function is the neural body, and the output means this is the action. Ultimately, what signal is coming out. So this takes input, it has importance factor, it calculates the contribution of each input to reach to our output. Now, the key components, already we have seen the inputs are there, the connections to neuron. Neuron is the basic input to receive the input. This is the basic unit, where this is our artificial neuron. These are all connections. Connections defined by the weight, that is the importance. The activation function gives, ultimately it maps the output. The value is mapped to the output level. The learning rule is a concept, that method that adjusts the weights and bias over time to improve accuracy. Yes. So this learning rule is the method that adjusts this weight and bias iteratively. That was done in our logistic regression also, in our classification methods also. It is a iterative process to solving equation. In the logistic regression we have seen. So here also it is a iterative process. Each time we are having a output, it is used with our existing known output, and this learning is done iteratively. We will see in detail. So there is an input layer where input parameters are coming. There is some hidden layer where these calculations, the iterative calculation is done, and the output layer where ultimately the outcome is the end result. So this is basic structure of a neural network, where each and every circle is a neuron. Each and every circle in this hidden layer is a neuron. Input layer is just to take the input. Output layer is just to give the probability output in terms of probability 0 to 1. And inside the hidden layer, all are these neurons. And all are connected to everyone. So this is a completely connected neural network. Yes, ma'am. Yes. So in this structure, we have to have the Excel sheet only in our hand. The similar way. Yes. I will discuss with some example, ma'am, for tabular data, for imaging data. So the basic structure is this, and how the machine learns. First, the forward propagation take place, means all inputs are given, calculation is done, and some output is there. The true value will be compared with the prediction value. Here, the loss function is the analysis of difference between the predicted value and the true value. Some errors will be there if it is true, but prediction may be false. true value, the negative for the negative class, it may be positive class. So false positive, false negative cases are there. The same kind of truth table, the false positive, true positive, false negative, true negative. Same thing is done here. The loss function calculate the amount of loss, and then that loss information is propagated backward. It is called backward propagation. And with this loss information, the model adjusts the weight and bias in the next calculation. So it is a iterative process until the loss function is minimized, the error is minimum. Yes. Yeah, we don't know how it is done, ma'am. It is a total black box things. Only we can understand these are the parameters and these are the output. For neural network, this is the problem. So in deep learning, for a simple neural network, when we go for a deep learning means there are lot of hidden layers and there are lot of calculations. How the weights and bias are adjusted, we cannot perceive. That is called black box. That is why it is called black box. Everything is inside. So neural network can handle the nonlinear data, means if this is a input data and it has a shape of, means the line, this blue and orange line is showing the characteristics of the data. It is not a linearly separable data, it is a nonlinear. And hidden layer, what it does, it gives different representation of the data, like SVM, support vector machine, and ultimately the nonlinear data can be classified. The neural network handles this nonlinearity very nicely compared to the machine learning methods when the number of data is huge and the pattern, the complexity is very huge, because it. Yes, sir. So there is code, Python code is there, and even having the Python code also, how the weights and bias are adjusted, that is encapsulated in the program itself, that we also cannot see. No, sir. The structure is same. That is the beauty of the thing. That structure is same. Little bit changes is done with our experience. How we will. Yes. Yes, sir. Yes. Yes, sir. So like the hyperparameter we saw in last class, there is some program. In the program, we can define the hyperparameter, means the parameter associated with the machine learning models. For neural network also, there is hyperparameters, and that we can adjust depending upon our experience, how we will adjust the hyperparameter to have a better accuracy. That we can do. Yes. I'm coming to that, ma'am. Yes, yes, ma'am. When we have a simple structured data, Excel sheet, machine learning is good enough. But when it comes, yes, when there is, yes, ma'am, yes, ma'am. When there is image, and we don't want to do that lot of work on that, having the ROI, extracting the features by ourselves, whole feature engineering workflow, then we can use that in the deep neural network that can handle image data. Yeah, I'll go there. And when another thing, when we have temporal data, we have for a patient baseline data after 2 days, after 3 days, after 5 days, maybe in ICU, or in course of treatment, maybe after 1 month, after 2 months, after 3 months, after 6 months. There is a temporal relationship for our clinical parameters. That is very well identified, the pattern, by neural network. There is a temporal deep neural network that can handle the temporal relationship between data. There the deep learning is useful. But if you have only Excel sheet for clinical features, then machine learning is good enough. Longitudinal. Yes, ma'am. Longitudinal. So deep learning is there when there is image input. I have shown here what are the exact data where we will go for deep learning. If there is image input, if there is temporal input, if there is multimodal input, when we have to club multimodal input, means clinical, image, pathological, maybe genomics also. So this is whole variety of inputs that cannot be handled by a simple machine learning model. Then this deep learning structure can come. It can separately handle separate data. Ultimately, it go to the end result. For different data, there is convolutional neural network for image, transformers for texture, generative adversarial network for generating our artificial image with our existing data if we need more data to generate, for recurrent neural network that handle this temporal data. Autoencoder is there to extract features. It is an unsupervised thing. And graph neural network, it can identify very complex relationship pattern. So these are very prominent deep learning methods that are used in healthcare. For different data, there is different architecture of deep learning methods to handle the data. So it is specific to the data. Basic building block is neural network, the artificial neuron, but how they have combined and they are analyzing data, that is different. So there are different structure, architecture. When we use some data set, we apply according to our data type. If it is image, if it is temporal, if it is text, or if it is, we need to generate more image, then this GAN is used. So according to our image or text or the data type, we will go for a different deep learning architecture. So these are all different architecture of deep learnings. If you don't have image, only text, then we don't use this. Only machine learning is enough, ma'am. If we have to use image, go for deep learning. If you have to image multimodal, image, clinical data, genomics, everything, go for deep learning. If we have unstructured data like medical records or OPD cards, then also the text analysis, this transformer can be used for that. So the data complexity has increased when the deep learning is the solution for that. So now we will see how image is handled. It will take time. Hmm. Yes. So when we have image data, we use convolutional neural network. This convolutional term is the specific term in computer vision for analyze a image to have the feature, to understand the image. What we see using our brain, the to identify a certain thing in the image. If you see an image, what you see, what is the object present in there, how the object is looking, what is the shape, means we don't think about it. But actually, when we learn, our brain learn how to see a scene, we see different components. We identify objects by their outlines. We can differentiate with the specific features like cat and dog, we can differentiate. That is inside our brain. We may not be able to explain it, but there is some understanding how we can differentiate things and we can see things. So to understand computer, to have that type of understanding of the image in terms of intensity only, the convolutional is the method to having the shape or structure or the spatial texture in the image. So how it is done, I will go further just to see how the convolutional neural network workflow works. Just to understand this, it is necessary. So CNN have the input as image. What image? It may be histopathological slide, it may be CT, axial or coronal slide, anything you can give. It is a image only. Input is image. Remember, for CNN, input is a image, not a tabular data, right? So we have a image. Every pixels have a value. That's all. The for a machine. Now filters are applied to input image to extract features like age, texture, and shapes. The whole thing of image interpretability lies in this convolutional layer. And for this filtering operation, the features is extracted and the feature maps generated by the convolutional layer are downsampled to reduce the dimensionality. This is pooling. First is feature map, convolutional is done, and then the pooled feature map is extracted. Means the feature is very diverse area. What feature is most important, that is pooled by this pooling layer. Means we are reducing our feature. We cannot handle thousands of dimension in to reduce the computer complexity. The computational complexity, we are just selecting some group of important feature. So the feature map is defined, extracted by filtering. And then the pooling layer is extracting the important features. Now we have the values, the tabular data. And now the neural network is handling the tabular data in terms of feature. To read the image characteristics, this convolutional layer that handles the feature extraction and that pulls the important feature, that is the extra thing in CNN. Now we will have just some idea how the features, means what it looks like. So convolutional layer, it has small matrix that is called kernel or filters. For example, it is an example. The left side matrix is our image. The small green square is a 3 by 3 matrix is our kernel. And it will go through the image sequentially and it will calculate some value. For it is an example, it will first attach to the left top corner and it will calculate some value and the center pixel will be replaced by the operation. This is called filtering. To some extent, I'll have some slides to explain it. How our image is a very big image. Kernel is a very small image. The red square area is a kernel. The black matrix is our image. So it fits on the position successively, pixel by pixel. Every position is covered by the kernel. And some calculation is done to have the features in the image. How it looks like? For example, if there is image intensity value in this yellow area and the kernel have 1 by 9 value, then what it is doing actually, it is taking the average of this value. If we multiply, this is a place by place multiply. Hmm. Yes. Hmm. Yes. Yes. Yes, 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 sir. So the yes, sir, the highlighted thing in the image will be more highlighted. The features means what is the characteristics in the image that will be highlighted, very prominent, so that machine can differentiate based on that highlighted areas. So what to look into it. So when we have this is some filter, and see how the after filtering the image looks like. Some anatomical area outline is now very prominent, and now they have very abstract kind of appearance, not very smooth city intensity type of thing. Now it is very abstract kind of thing. This is very high level of uh, interpretation filtering. Now see, this is another type of filtering. In the image, some area, some horizontal type of uh, lines or vertical type of lines or some mixture kind of pattern in the intensities that is captured. So means in the image, if we see very abstractly how the how it is looked, what are the prominent features, prominent characteristics in the image, that is the overall characteristics of the image. And it has lot of different type of filter that is used, and for different image type, means for histology image, for ultrasound image, or it is a endoscopic video image. For different type of image we use different filtering methods. It's a whole different area in computer vision, how we use filtering to have the prominent features in the image. So depending upon features, this filtering is also it changed, and the image information is captured. So pooling layer is just having the most important thing. If it is max pooling, having the maximum value. If it is mean, having the mean value in the area. Very simply, we are down sampling the numbers. The features maybe when we apply a deep learning based feature extraction, it is in thousands, 2000, 3000 features. We cannot understand what is the value it means. That is why it is black box. But the features, the characteristics is identified with these filtering techniques. How computer is understanding, that is not understandable by us. And then it is pulled. The important features is pulled. No, we will give only image. Machine will do everything. Outcome means the classification, the risk stratification. So the applications may be classification, detection, segmentation. Area segmentation or detection means just telling a bounding box, here is the tumor. Segmentation means identifying the whole area of the tumor. There may be semantic segmentation of, like in cellular image, the different type of cell can be also differentiated after segmentation. So these are all different applications. And if we summarize everything, we are having an image input. That's all we are giving. The whole next, everything is done by the deep neural network, CNN, convolutional neural network. First, it is having low level feature extraction. That is the edge detection and the blob detection. Blob means some gross area, rounded or structured. Edge means the fine lines, the prominent lines, maybe horizontal, maybe diagonal, maybe in other directions. So these are all very prominent type of information that we have seen in the filtering method, right? So then we have the second level of mid level of feature, that is called textural features, that I was telling in terms of spatial orientation. And then the deep feature composite, the anatomical context is identified, where the position, the means when we are seeing a liver, the hepatic changes or liver tissue changes, that is totally different anatomy. So that context is also understood by the deep neural network, by its positioning and with the features. And then all features go into the fully connected layer, where the weights and biases are adjusted and the features are ultimately mapped for the classification or detection or segmentation task. So this is the whole pipeline of a CNN analysis. Input is only image. That is the beauty of it, but we don't know what is going on inside. The explainability is a process to identify exactly which area of the image is giving us the classification or detection result. That is a process that we'll see in later classes. How we do explainability to understand deep learning analysis. Because everything is black box, we cannot understand, and for the reliability purpose, clinical explanation has to be developed to believe what it is giving, it is doing correctly. So that is another aspect. So for now, just understand this is the whole workflow for a convolutional neural network, when input is only image. Here is some example for Rakhi ma'am that you were asking, with some example how we give the input and what we have at the end. So here the input is image. The two level of deep learning method is happening here: localization and classification of common hepatic lesions. First, the CNN segment the liver, only input is image, axial CT images. Then the first CNN segment the CT liver axial area, and then the second shaded model that localize the liver lesion in the slice. So here you can see what is the in the workflow. Everything is CNN model only. We are having yes, yes, by machine only. We don't need to do the area of earlier in the radio mix when we do the feature engineering by hand, means ourselves. Then we have to draw the ROI. Here we don't need to draw even the ROI. Yes, you identify whether there is some tumor. Yes, 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 ma'am. So these additional things may be after identifying this tumor in the image, this tumor can be this image portion, the identification portion can be analyzed to have a risk stratification, to grading the fibrosis, or to having the survival prediction. uh, Depending upon the tumor texture, we can predict the maybe long-term survival. Mhm. Yes, yes, yes. So for that case, the some example of problem may be to differentiate the fibrosis level, to differentiate the steatosis level in the histopathological image. That is very subjective. Yes. Yes. 100. Yes. Yes. Necrosis. You have to feed all that. So, first we have to train the machine with the given images and we have to supervise learning. Yes. So, super deep learning is the supervised learning here, sir. We will give three set of image with the labeling. This is fibrosis, this is tumor, this is necrosis. 100 image we given. Then the model is trained and it will have some training accuracy level and all. Then we can give a new image. It then can tell whether it is fibrosis or necrosis. Yes, because it is seen by the image. So so when we are doing the application like screening, whether a image is normal or abnormal, means there is some pathology, that type of screening can be also done. Means for a yes. Mm. So that level of subjectivity is there, but we can use consensus from multiple pathologists, and we can use consensus labeling to make the subjectivity minimal, and then we can train a model. Then every time it will give the same assessment. So in that way, that process of subjectivity can be to some extent eradicated. So that type of thing that is repeated and subjective, that can be applied using deep learning. But where when the clinician itself is a good enough to tell what is the means to assess the diagnosis, then it is not required. But when we are doing very repeated thing and it is time consuming, maybe it's for screening whether it is healthy or whether it is not healthy, or us grading seeing the histopathological image, there may be lot of patches from a single slice. So lot of patches takes lot of time. So if we have a automated thing to just to have a assessment, then that can be checked after the assessment by CNN and confirmed by the pathologist. So this type of workload distribution can be reduced. Yes. Mm. Yes, yes. Yes, the basic. Yes. So, yes, it is possible to do. We, yes. Y- You again, you have to tell machine learning will be done by your decision levels. So, in that case, I will ask, sir, the pseudo progression means the cell proliferation is not tumorous, not malignant. So, so if that area, if that is for human eye, it looks like tumor, but for computer, if we do the texture analysis, it is uh, this type of application is available. Yes, yes, that type of system is already developed, and one this type of work already I have done, and that project is in multi-center level actually, ICMR funded project. So, so the texture analysis. Yes. Yes, yes, sir. So the tissue characteristics, the intensity level itself tells the tissue characteristics. So that is the radiomics thing. The texture can tell whether it is a tumor tissue or not. It is a pseudo progression. Yes. No, no, no. Multiple RUI. Any. Mhm. Mhm. Hmm. That machine can does it, and multiple lesion can be processed at once, like in lung CT, where nodules are at everywhere. So each and every nodule can be segmented, each and every nodule can be processed with the radiomics analysis, and each and nodule can be told about whether it's a benign, malignant, or a metastasis. No cytostasis. It is a kind of digital pathology. You can tell. Yes. So that is our ground truth, sir, clinical ground truth. But what is deep learning is doing to reach to towards that to make the thing automated, easier, less time consuming. So always we have the clinical ground truth is the pathological sample only, analyzing the histopath. But this is the way to do to shorten the thing to make the clinical load reduced. Basic screenings tool. It is not the ultimate thing. We cannot rely on it wholly, like histopath. So this is from the beginning. I am telling this is not something you can rely 100%. Yes. See, these all these things are evolving area, sir. Everything is not available for use, clinical use. Many things are under research. So these are all possibilities. We can work towards these areas. Yes. Well, there is lot of applications available in clinical domain that is in use actually, and some areas are still in progress. Okay. So, So in that area, ma'am, I want help from you actually. What clinical applications we want to discuss that if that comes from clinicians, that will be helpful for me. Yes, sir. Hmm. So recently, that work I have done with Dr. Harsh to identify the mortality levels risk using that CT image in hepatic encephalopathy. Uh, this almost 50% is again we have to go in means the transformer text. Thank you, sir. Sir, it is going to be difficult actually.